Welcome back to Cancer as a Mitochondrial Metabolic Disease Basics playlist. When you eat carbohydrates, your body turns them into glucose. But then what happens? Before your cells are able to make ATP or cellular energy efficiently, even before the mitochondria get involved, there is a critical first several step process, and it's called glycolysis. In this video, I'm going to show you how cells break down glucose, why this pathway exists, and why understanding it is essential for us to talk about before cancer metabolism and mitochondrial dysfunction. So without further ado, let's get into it. So I have been really trying to think about what I want to do with this cancer as a metabolic disease basics playlist because this is going to be mostly for probably a little bit of a different audience. And because on the main playlist, we go into very advanced, high level topics, but it assumes in some ways that you have at least a good understanding about metabolism. Now, a lot of the concepts that we go over are repetitive, but in this particular playlist, I'm going to go over some things that were really never directly covered on this channel before. And so I think before we talk about ketosis and amino acid fermentation and kind of the nuances around that, I think that we need to probably introduce some of these more basic concepts. And I'm going to try to do it in a succinct way that doesn't bore us with super technical biochemistry, but at the same time, helps us understand what the reason is for these pathways and the fact that they exist. So I'm going to start out with talking about glycolysis. So I'm going to use some kind of National Libraries of Medicine resources to help guide us. And then I'll use some other papers to help kind of like add context to why we should even care about this. So this was published in Stat Pearls, Biochemistry, Glycolysis. And it says glycolysis is a metabolic pathway with an anaerobic energy source that has evolved in nearly all types of organisms. Another name for this process, and actually funny enough, Dr. Seefried a lot of times refers to it as this, is as the Embden-Meyerhof pathway, in honor of the major contributors towards its discovery and understanding. Although it doesn't require oxygen, hence its purpose in anaerobic respiration, it is also the first step in cellular respiration. The process entails the oxidation of glucose molecules, the single most crucial organic fuel in plants, microbes, and animals. Most cells prefer glucose, although there are exceptions such as acetic acid bacteria that prefer ethanol. In glycolysis, 2 ATP molecules are consumed, and it produces 4 ATP molecules, 2 NADHs, and 2 pyruvates per glucose molecule. The pyruvate can be used in the citric acid cycle, which we'll talk about in a later video, and serve as a precursor for other reactions. It says here that glycolysis ultimately splits glucose into 2 pyruvate molecules. One can think of glycolysis as having 2 phases that occur in the cytosol of cells. The first phase is the investment phase due to its usage of 2 ATP molecules, and the second is the payoff phase. These reactions are catalyzed by their own enzyme, with phosphofructokinase being the most essential for regulation as it controls the speed of glycolysis. Glycolysis occurs in both aerobic and anaerobic states. We're going to kind of differentiate the two between aerobic glycolysis and anaerobic glycolysis. In aerobic conditions, pyruvate enters the citric acid cycle and undergoes oxidative phosphorylation, leading to a net production of 32 ATP molecules. In anaerobic conditions, pyruvate converts to lactate through anaerobic glycolysis. Anaerobic respiration results in the production of 2 ATP molecules. Glucose is a hexose sugar, means 6 carbons, meaning that it is a monosaccharide with 6 carbon atoms and 6 oxygen atoms. The first carbon has an attached aldehyde group, and the 5 other carbons have 1 hydroxyl group each. During glycolysis, glucose ultimately breaks down into pyruvate and energy. A total of 2 ATP is derived in the process: glucose plus 2 NAD plus 2 ADP plus 2 pi inorganic phosphate equals 2 pyruvate, 2 NADH, 2 hydrogen plus, 2 ATP, and 2 water molecules. The hydroxyl groups allow for phosphorylation. The specific form of glucose used in glycolysis is glucose 6-phosphate. At the cellular level, glycolysis occurs in the cytosol of cells. Under aerobic conditions, pyruvate derived from glucose will enter The mitochondria undergo oxidative phosphorylation, which we'll talk about later. Anaerobic conditions result in pyruvate staying in the cytoplasm and converted to lactate by the enzyme lactate dehydrogenase, or LDH. So I always like pictures better. So we've talked about a lot of text. Let's look at some pictures. So as the pathway implies, this is the breakdown of glucose. So we're going to start with glucose, the 6-carbon glucose molecule right here. There's going to be a series of steps, and we're going to end up with some important kind of end products, okay? So glucose, these are the intermediates: glucose 6-phosphate, fructose 6-phosphate, et cetera, et cetera, et cetera. The steps are not that important for our level of understanding. These annotations over here, hexokinase, phosphohexoisomerase, phosphofructokinase, et cetera, are the enzymes that convert glucose as glucose 6-phosphate and glucose 6-phosphate to fructose 6-phosphate, et cetera. Those also are not critically important to know this second, although we have talked about them in the other series about areas where we could potentially use either drug or nutrient inhibitors of these as a way to employ metabolic therapies. But my point is that this is a 10-step process that is critical for the breakdown of glucose. And the end product of this is pyruvate, and pyruvate under normal conditions would get into the mitochondria and then go through the TCA cycle and the electron transport chain. However, if it is not getting into the mitochondria for whatever reason, and it's not going to get broken down by the mitochondria, then pyruvate could also be converted into lactate. Here's the lactate dehydrogenase enzyme it talks about, LDH. And I also pulled this because this is kind of an interesting thing. So this is the investment phase they're talking about, and this is the payout phase they're talking about. So this is where we invest 2 ATPs and we get 4 back for a net of 2. And then this is just another picture showing the investment phase. Here's our -2, here's our +4. Overall, 2 net ATP, 2 NADHs, and then we get the 2 molecules of pyruvate, like it said in the text. And I'm going to next use this paper to help us understand even more deeply why this is important for overall health, but also for cancer specifically. So this paper was published in November 2024. It's titled Glycolysis, a multifaceted metabolic pathway and signaling hub. It says here that glycolysis is a highly conserved metabolic process responsible for the anaerobic production of ATP, adenosine triphosphate, from the breakdown of glucose molecules. While serving as a primary metabolic pathway in prokaryotes, that's not us, glycolysis is also utilized for respiring eukaryotes, that is us, providing pyruvate to fuel oxidative metabolism. That's when we have normal mitochondria, that's when we have oxygen present, that's when we're under normal conditions. Furthermore, glycolysis is the primary source of ATP production in multiple cellular states, hypoxia, and particularly important in maintaining bioenergetic homeostasis in the most abundant cell type in the human body, the erythrocyte, the red blood cell. Beyond its role in ATP production, glycolysis also functions as a signaling hub, producing several metabolic intermediates which serve roles in both signaling and metabolic processes. These signals emanating from the glycolytic pathway can profoundly impact cell function, phenotype, and fate, and have previously been overlooked. In this review, we're going to discuss the role of glycolytic pathway as a source of signaling molecules in eukaryotic cells, that is us, emphasizing the newfound recognition of glycolysis' multifaceted nature and its importance in maintaining cellular homeostasis beyond its traditional role in ATP synthesis. And let's take a look at this graphic that's associated with this paper. So we have glucose, we have the 10 steps, we have the things we looked at in that first couple of slides, but we looked at it only from an energetic perspective. What this paper is going to talk about, at least very basically, is that it's not just about energy. It's also about other things. So you can see here that, for example, 3-phosphoglycerate can then be converted into making amino acids, or glyceraldehyde 3-phosphate can go towards making lipids. Also, amino acids can go towards that as well. Or glucose 6-phosphate can be used to make glycogen or nucleotides, DNA, RNA, or glucosamine, and other kind of complex sugar molecules. And then it's not apparent here, which we're going to have a whole video on the PPP, but this is part of what they call the pentose phosphate pathway, and that's important for redox homeostasis, or the balances of antioxidants to oxidants. And then, of course, there's always energy, and there's feedback loops, and it gets very complicated. But I just wanted to give a little bit of a higher level understanding of why glycolysis is important. So it says here, the glycolytic pathway is a highly conserved metabolic process responsible for the anaerobic catabolism of glucose with the concomitant production of ATP. The pathway consists of 10 metabolic enzymes, which work in a sequential manner to convert a 6-carbon glucose molecule into 2 3-carbon molecules of the tricarboxylic acid TCA precursor pyruvate. The anaerobic conversion occurs within the cytoplasm and results in a net gain of 2 molecules of ATP, 2 molecules of reduced nicotinamide adenine dinucleotide, NADH, and 2 molecules of water per molecule of glucose entering the pathway. Under physiologic tensions, the pyruvate generated from glycolysis is transported into the mitochondria where it undergoes O2-dependent oxidation to form acetyl-CoA. The acetyl-CoA is metabolized by the TCA cycle and the electron transport chain during oxidative phosphorylation to produce a significantly greater amount of ATP per glucose molecule than produced from glycolysis alone. While glycolysis serves as the initial step in cellular respiration in most respiring cells, it plays a crucial role as a primary source of ATP for cells lacking mitochondria, like erythrocytes or cancer cells, or the cells experiencing hypoxia, again, cancer. Under such conditions, a significant increase in glycolytic flux is essential to maintain ATP production and bioenergetic homeostasis when mitochondrial metabolism is reduced or absent. The increased flux is supported by the fermentation of pyruvate into lactate, a process which concurrently regenerates NAD, an essential cofactor for the sustained glycolytic flux from its reduced form NADH. Cellular respiration encompassing both glycolysis and mitochondrial respiration is a highly regulated process. This precise regulation allows for efficient energy production while permitting rapid adjustments to the rate of respiration to be made to ensure that dynamic energy requirements of the cell in a changing environment are met. While we have predominantly discussed the transcriptional role of glycolysis and its effects on cell metabolism, this review will focus on our current understandings of the role of glycolytic pathway as a signaling source in eukaryotic cells, which again is us. So I'm going to stop there because I think it's plenty of meat to chew on during this basic series. But basically, what the point of this video is to talk about there is a process that your body uses to break down glucose once we eat carbohydrates. Okay, that process is called glycolysis. There's 10 steps. It requires enzymes, it requires cofactors, and it can be done under aerobic conditions or it can be done under anaerobic conditions, basically with oxygen present or without oxygen present. And there are two phases, basic phases. There's the investment phase where you have to actually invest ATP or cellular energy to get more energy back. You get a net 2 ATP, and then under aerobic conditions, that pyruvate is going to go into the mitochondria and it's going to undergo oxidative phosphorylation. And under anaerobic conditions or conditions without oxygen or where mitochondria are damaged, you're going to end up with fermentation from pyruvate into lactate. You're going to see how this is critical information to understand cancer metabolism as we talk about aerobic glycolysis or the Warburg metabolism in future videos. If you like videos like this, please like, share, and subscribe. Until next time.